Hello students, welcome back. Previous two videos we were attempting to explain the events that occur in the contraction and also the events that occur in excitation. This time around we're going to try to put them together and close any loops, and we're going to do it by putting each set of reactions together in what is typically known as the excitation contraction coupling reactions. And when we take a look at these, start back at where we just finished, the excitation. Remember, the electrical event comes down the surface of the axon, heavily myelinated, comes to the axonal terminal. Here we have voltage regulated calcium gates. Those gates open, calcium floods in. When calcium floods in, it stimulates the synaptic vesicles that have acetylcholine to migrate to and fuse with the axonal terminal, releasing the acetylcholine into the synaptic cleft here at the neuromuscular junction. The acetylcholine migrates across, if you recall, binds to chemical regulated sodium channels or gates, opens up those gates, sodium comes flooding in. We get depolarization locally, which spreads, if you recall, as that sodium comes in here. It's a localized depolarization, but remember, sodium ions do not like each other, so they repel each other. So it's a bilateral event. Remember that sodium goes both ways. There'll be sodium channels here that are voltage regulated channels, and this electrical event will just repeat itself all the way down through here until it comes to our triad. Now here's that triad. Remember when we were in anatomy portion of the muscle, here we have the terminal end of a sarcoplasmic reticulum. Here we have another terminal end of the sarcoplasmic reticulum. In between those two is the transverse tubule or the T-tubule. That electrical event that's coming down the surface of the sarcolemma will dip down into these. It'll also skip over it, as you see, but it'll dip down into it. And we'll talk about the fact that it'll have an impression upon some surfaces here that will stimulate the release of calcium. Now, when that calcium is released, remember its role is to go and bind to the troponin. And when it binds to that troponin, it causes the troponin to change shape in such a way in which it moves the tropomyosin out of the way so that we can have that cross-bridge attachment and we can have the contractions occurring. Now, the only thing we really haven't looked at is what is the role of the triad. So here again, from that view you saw, the action potentials coming down the sarcolemma until it gets to this complex we call the triad. Again, that's the triad comes from the sarcoplasmic reticulum here, another one over here. These are the terminal ends. And then this transverse tubule that dips down into the muscle fiber itself. That electrical event action potential comes down through here as well as skipping over here. When it comes here, there are some voltage regulated gates here. Now these voltage regulated gates are typically made up of four what's called foot proteins, because what proteins do is hold things together. In this case, the proteins are holding the end terminal of the sarcoplasmic reticulum, fusing it basically to the T-tubule. And when the electric event comes down, these are voltage sensitive tubule proteins, and these proteins will open up and allow the release of calcium, and calcium will flood into the cell itself. This is called an efflux, E-F-F-L-U-X. And when the calcium goes in, remember what the calcium does. Calcium goes in, it binds to the troponin component, causes a change in the troponin in such a way that it allows the tropomyosin to slide down, exposing the myosin head group binding sites on the active molecule, and we have the first cross bridge attachments. Now let's put all that together. Here what we see again is we see the electrical event one last time. Action potential goes down the surface, skips over here, but also comes down the triad. As it comes down the triad, it triggers open the voltage regulated calcium gates. Calcium comes flooding into the muscle cell. Calcium binds to the troponin. As more and more calcium is flooded in, more and more troponin molecules are altered their conformation as the calcium binds to it. Thin filaments are exposed, more and more cross-bridge attachments occur. So the more stimulus we got, the more frequent stimulus we got, the stronger the stimulus we got, the more calcium that's going to be present in the muscle. the more potential to have a stronger and stronger muscle contraction. Because remember, the sum total of the muscle contractions is directly correlated. The calcium we got there inside the cell, the more muscle contractions we can have. Pretty neat to think the calcium does that, because most people think it only is used for bone. Now, since we talked about the excitation contraction coupling events, there are a series of things that must go right for all those events to occur. I think you got to agree with me there, but one of the key components is we always have to have ATP. ATP, ATP, ATP. It's the energy that we need to make sure the muscle contraction occurs. And I hope you see where that ATP is needed at in this event. It's needed in several places. Frequently, you would think of them as pump areas, but remember at the axonal end, calcium comes in to stimulate the release of acetylcholine. Well, as soon as that electrical event stops, we want to get that calcium out, because any buildup of calcium in that axon is going to result in not being able to stimulate the release of acetylcholine the next time there's an electrical event coming down. So we actively pump the calcium out at rest. That takes ATP. If you remember on the postsynaptic end of the the synaptic cleft there in the neuromuscular junction, we got those sodium-potassium gates. Remember, sodium comes in, potassium goes out. Well, we can close those gates. There's still going to be more sodium on the outside than there is on the inside temporarily. But if we did this repeatedly without pushing the sodium back out, pumping the sodium back out, at some point I'd open those gates and sodium would move out instead of move in, because remember, it's simple diffusion. It's allowing this movement to occur. So we got to actively pump at rest the sodium that moved into the cell there at the neuromuscular junction back out, so we have that, we can reestablish that resting membrane potential. Another place that you're going to find it at is associated with those sodium-potassium gates that are found along the length of the muscle that was associated with the action potential. You're also going to find it down in the triad. Remember, we opened up those voltage-regulated calcium gates and allowed calcium to flood out. Typically, the thing that stops the muscle contraction is the same thing that initiated it, except the reverse. We got this concept called negative feedback mechanism, if you recall. Negative feedback mechanism says, I'm thirsty, I drink, I'm not thirsty anymore. Well, in this case, I need a muscle contraction. I release calcium. I've done the muscle contraction. Now I need to stop the muscle contraction. I got to take the calcium back in. Anytime I open those gates and allow calcium out at the triad from the sarcoplasmic reticulum, it's a good bet there's still going to be more calcium when I close those gates than there is in the sarcoplasmic reticulum than there is in the muscle cell itself. So anytime I'm taking something from a lower concentration in the muscle cell to a higher concentration in the sarcoplasmic reticulum, I'm going to have to use ATP to pump it back in. And then remember, the last place where you need a lot of ATP is once you've got that calcium out. Remember that whole process, those 4 steps in it of attachment, pivot, detachment, cocking the head group, attachment. Each time I go through one of those cycles, I use an ATP molecule per head group. Now there's millions of head groups that are doing the same thing. So imagine all the ATPs it takes just to cock the head groups. And remember, I'm going to do that as long as the head group is capable of binding to the active molecule. I'm going to cock it, or until I run out of ATP. And at that point, I might have a muscle cramp. So now, it is your time to take a deep breath, do some review of your muscle anatomy, tied into your muscle physiology as associated with the contraction events, the excitation events. And then when you start looking at the coupling, don't forget that the coupling event there is when we discussed the triad, because we had the excitation, we had the contraction components, but where does the rubber meet the road? The rubber meets the road when the excitation can trigger the release of calcium that can then trigger the contraction reaction. So remember, something as simple as calcium ions are the trigger associated with a muscle contraction being capable.